Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Cell and Molecular Biology, we are discussing about the different aspects of the molecules, how they are interacting with each other, and how these interactions are resulting into the different types of processes. In this context, so far what we discussed about the origin of life, we have discussed about the evolution, we have discussed about the cellular structures, whether it is a prokaryotic structure or the eukaryotic structures. And then in the previous lecture, we have discussed about why the growth is very important for the different types of organisms. So we have discussed about that the growth and the reproduction is the fundamental aspects of any living organism. So there are discrete properties of a living organism. It should be responsive to the different types of stimuli. It should be very having the complex organizations, and the most important property of a living organism is that it should be able to have the growth potentials, and these growth potentials should be endogenous rather than exogenous, and then it should be able to reproduce and give rise to the offsprings. Now, in the previous lecture, we discussed about the growth, how the different phases of the growth are present in the prokaryotic system and as well as the eukaryotic system. What are the different places where it can be controlled so that you can have the growth in a very tight controlled processes. And we have discussed about how the growth is being controlled at different stages in the prokaryotic system, and what are the different cellular machinery what is required for controlling the cell division within the prokaryotic system. And then we also discuss about the eukaryotic system, and we have discussed about the different stages where the growth can be controlled. Now, when you talk about the growth is very important. And if you recall, in the previous lecture we discussed about that for the growth, the energy is very important, and because the energy is required in terms of driving the so many interactions, whether it is being done by the ATP and the ATG complexes and all that. So when you talk about the growth requirements are very different. We have discussed about the prokaryotic cell, we have discussed about the eukaryotic cells, and within the eukaryotic cell we have also discussed about like animal cells, plant cells, fungi, yeast, and so on. So when you talk about the growth, every organism's requirement for a growth is very different, and this is the topic what we are going to discuss in today's class, and how the different organisms are acquiring the nutrition or for their growth, and what are their requirements. So when we talk about the growth media is actually a complex material, right, or complex solution, and the main purpose of a growth media is to provide the essential biomolecules or essential nutrients so that it could be able to allow the organism to build the basic biomolecules. When you talk about the basic biomolecules, you are going to talk about the proteins, you are going to talk about the carbohydrates, you are going to talk about the lipids, and the nucleic acid in form of the DNA and RNA. So when we talk about the growth media, so growth and the multiplication of organism used as an expression system require a suitable biochemical and biophysical conditions. The biochemical, that is the nutrition condition, can be provided by the use of the various nutrient media. Depending upon the special need, there are different types of media have been developed for expressing to the different achieve the growth, multiplication, and the overexpression. So when we talk about the growth media is required for the organism to build its building block, right? And the building blocks are protein, carbohydrate, lipids, and the nucleic acids. When we talk about the carbohydrate, or when we talk about the proteins, the protein is being made up of the amino acids, and amino acid is the building block for and they are actually working as a building block, which means the proteins are mainly being used for making the different types of structures. For example, the plasma membranes, protein is being used for making the multimeric protein complexes so that they can be able to participate into different types of reactions and so on. So their main job is to provide as a building block, right? And the proteins are made up of the different types of atoms such as carbon, hydrogen, oxygen, nitrogen, and the sulfur. Similarly, for the carbohydrate, the monomeric unit is sugar, right? It could be a monomeric sugar such as glucose, fructose, mannose, and galactose. And then these sugars are being burned into the different types of carbohydrate metabolic pathways such as glycolysis, Krebs cycle, and so on. And using that metabolic pathways, the sugar molecules are being used for producing the energy. So the main purpose why you require the carbohydrate, why the organism require carbohydrate, because it wants to produce the energy. And the carbohydrates are mainly being made by the three atoms such as carbon, hydrogen, and the oxygen. There are carbohydrates where you actually have the other atoms also, but that proportion is very minor, such as you have the chondroitin sulfate and some those kind of carbohydrates, but they are specialized carbohydrates. They are not being used. They are not being used for the energy production. Similarly, we have the lipids. The lipids are made up of the fatty acids, and these fatty acids are also being used for producing the energy. And fatty acids are, or the lipids are made up of the different types of atoms such as carbon, hydrogen, oxygen, phosphorus, and the sulfur. Then we have the nucleic acid, and nucleic acids are made up of the nucleotides, and nucleotides are being used for carrying the information from the one generation to next generations. And this anyway we are going to discuss in detail when we are going to talk about the genomic DNA or the genomic content of the organism in a following modules. And the DNA and RNA are made up of the carbon, hydrogen, oxygen, nitrogen, and the phosphorus. So what you see here is that even if you have to synthesize the proteins or the carbohydrate, lipids, or the nucleic acids, you basically required a nutrient source which can be able to provide you the carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus, and the nitrogen. So basically require a nutrition source so that you can be able to provide these nutrients or these atoms actually. This can be provided in a complex form or it could be provided in a simpler form. Apart from these requirements, you also require a very small quantity of minerals, and you also require the different types of vitamins which are actually going to function as a coenzyme. So considering these requirements for the organisms, people have developed the different types of microbiology media or different types of media for the different types of organisms. So let's first talk about the microbiology media, and then we will talk about the eukaryotic media. So when we talk about the microbiology media, remember that when you are going to provide any media, your prime objective would be that the media should be able to provide these atoms, right? So that you can be able to use these atoms to synthesize first the monomers, and then these monomers will come together to give you the polymer or the complex molecules, right? So for example, if you use these atoms, they are actually going to be used for synthesis of the amino acids, and then these amino acids can be used for synthesis of the proteins by a very complicated process of translation, right? And same is true for the nucleic acid also. So when we talk about the microbiology media, right, you are actually going to use the different types of constituents. So you are going to use the amino nitrogens, you are going to use the growth factors, you are going to use the energy source, you are going to buffer, you are going to different types of minerals and metals, selective agents, indicator dye, and gelling agents. So when you require the amino nitrogen, which means this is actually going to provide the nitrogen for the organisms, you can actually be able to use the peptones, protein hydrolysates, infusions, and extracts. Similarly, the growth factors, they are also required, so you can actually be able to use the blood, serums, yeast extracts, vitamins, or NAD. Then you require the energy sources that actually is going to provide the carbon, and for that you can actually be able to use the sugar, alcohol, and the carbohydrate. Then buffer is required because we are using the in vitro system, so the pH should not change. So for that, you can actually be able to use the phosphate, acetate and citrate. Remember, these phosphate, what you use for the buffer is also going to be a source for the phosphate, right? Then you are also going to use the mineral salt and metals. So you can actually be able to use the phosphate, sulfate, magnesium and calcium and iron. Then you are going to use the selective agents. Selective agents are going to be used for the selection of the organisms. So that may be having the application in the recombinant DNA technology, where you are going to use the selective agent for selecting the transformed cells from the untransformed cells. So for that, you are going to use the antibiotics and dyes and chemicals. Similarly, you are actually going to use the indicator dyes so that you can be able to see whether the media is good or bad. For that, you are going to use the phenol red or the neutral red. And ultimately, you are going to use the gelling agents. Gelling agents are going to use for converting a liquid media into a solid media. So you are going to use the agar, gelatin, alginate and silica gel. Now, considering these nutrient or these constituents which you are going to get from these complex substances, the people have developed the different types of microbiology media. So they have used the different types of media. So one is called as M9 minimal media, M63 media, LB media, SOB media and so on. And when you see the composition, right, for the minimal media, it is going to be sodium hydrogen phosphate, potassium hydrogen phosphate, sodium chloride and the ammonium chloride. And this is going to be used for the cultivation of the all sort of strains of the E. coli. Similarly, you have the M63 minimal media, where you are going to use the ammonium sulfate, potassium hydrogen phosphate base 6 and the ferrous sulfate, and that is you are going to use for the cultivation and maintenance of the E. coli. Similarly, we have the LB media, where you are going to use the 1% peptone, yeast extract and 1% NaCl, and that you are going to use for the E. coli growth, so that you can be able to prepare the plasmids and you can use this for the protein production. Similarly, we have the slightly derived version of LB media, and there also you are going to use that for the propagation of E. coli. Similarly, we have SOB media, where you have the 2% peptone, 0.5% yeast extract, NaCl and the KCl and MgCl2, and that is being used for making the high efficiency competent cells. Then we have the SOC media, and that is going to be used for making the competent cells. Then we have the YT media, that is for peptones, yeast extract and NaCl, and that is going to be used for the fast DNA productions. Similarly, we have the super broth, where you are going to use the 3.2% peptone, 2% yeast extract and 0.5% NaCl, and that is being used for the high yield of the plasmid DNA and the protein production. So all these microbiology media, some are minimal media, like some are defined media, some are very complex media. For example, the media where you are actually adding the salts and all those kind of things are actually being called as defined media, right? So where you are actually having the media component which are defined, so these are called as defined media, where you have the the defined amount of disodium hydrogen phosphate or potassium hydrogen phosphate, sodium chloride and ammonium chloride. Compared to that, for example, the LB media, it is not a defined media, because you have the peptone and yeast extract and NaCl, and peptone is actually a very complex biological material, right? Peptone is actually having lot of cellular material, right? So the composition of the constituents present within the peptone is not very under the control of the user, right? So that is the same thing is for true is for, like, for example, you have the M63 minimal. So all sort of minimal media what you use, they are actually the defined media, and defined media is being used in those cases where you are actually trying to optimize the different types of parameters, and you also want no contamination or any kind of interference of the complex biological substances from the microbiology media. So in those cases only you are actually going to use the defined media. Now, how you are going to prepare the media? So for preparing a media, we have taken an example of how you can be able to prepare the LB medium. For preparing a bacteriology media, dissolve the component as per this particular table. Like for example, LB media, you are going to take the 1% peptone, 0.5% yeast extract, and 1% NaCl, and then you define that into 1 liter of distilled water, right? Cover the top of the flask with the cotton plugs and autoclave the solution at 121 degrees Celsius for 20 minutes, right? So when you do so, you are actually going to make the microbiology media. Remember that you are using the autoclaving for sterilization only in the case of microbiology media, right? So while you are doing the autoclaving, you are supposed to take a lot of precautions, right? What are the precautions you have to take? Number one is the ratio of the media volume and the culture flask should be defined, right? For example, when you are making a 250 ml media, right, then it has to be prepared in 1 liter flask, so that you are actually going to have 750 ml of oxygen, right? So you are actually going to have the 3/4 oxygen, and that 3/4 oxygen is good enough to provide the aeration. Because remember that once you are going to prepare the media, for example, if you have taken a flask, right, you are going to put a cotton plug, right? You are going to put a cotton plug on top, right? Once you put the cotton plug, it is going to be disconnected from outside, right? So there will be no oxygen what it actually can take up from outside. That means the bacteria has to survive whatever is oxygen is present within this flask. That means you should have the sufficient ratio of media volume and culture flask so that you can be able to provide the enough aeration, so that this air is actually bacteria is going to use for their running the different types of metabolic pathways. Then media components are hygroscopic, and while weighing, you should avoid the moisture stored in a cool and dry place, which means all the media components are very dry, so that you can be able to add these media component in a defined conditions or defined amount. So you should be able to very careful when you are weighing these media components, so that you should avoid putting the moisture into the media component, and that's why you should store the media in a cool and a dry place. While autoclaving, open the autoclave only when it is cold. Remember that autoclaving media means you are actually going to autoclave something at 121 degrees Celsius, and at this temperature the water is actually going to be boiling, right? Even if when the autoclaving is done. So if you open the autoclave, this boiled water will actually going to spill, and it will actually going to be cause the injury. That's why you should be very, very careful. When it get cooled down, then only you should open the autoclave. Then you avoid the charring of the media component. So you should have the sufficient quantity of water within the autoclave when you are doing the autoclaving, so that there should be no charring of the media component. It means there should be no burning of media component. Then the solid media should be poured in a plate once it is cooled down. So when you are preparing the solid media, like you are actually can prepare the media in two ways. One is the liquid media, the other one is the solid media, right? Solid media where you are actually going to put the gelling material, right? So in that case, you should let the media to be get cooled down sufficient enough, so that when you add the antibiotic should remain within the solution, right, and should be active, right, and that is only possible when you cool down to around 50 to 55 degrees Celsius, and then only you add the antibiotics. The various antibiotics or nutrient supplements should be added to the media when the temperature is less than 50 degrees Celsius, right? So this is all about the how you can be able to prepare the microbiology media, and how you can be able to autoclave, and what are the different types of precautions you should take. So to explain these phenomena, I will take you to my lab, where my students are going to show you how you can be able to prepare the microbiology media, and how you can be able to prepare the cotton plugs, and how you can be able to perform the autoclaving.
In this video, we are demonstrating how to prepare bacterial culture broth. For preparing culture broth, we need three components. One is peptone, yeast extract, and sodium chloride. For 100 ml of culture broth, we need 1 gram of peptone, 0.5 grams of yeast extract, and 1 gram of sodium chloride. I'm going to weigh individual components and dissolve it in double distilled water. Then we have to autoclave the media. Before weighing, care should be taken. Spatula is clean and the balance is taken. After weighing, we have to clean the spatula and keep it in original position. And during weighing, care should be taken to avoid contact with any of these media components. After weighing the media components, we have to dissolve them in double distilled water. So initially, we are dissolving in 80 ml of distilled water. Once the components completely dissolved, we have to make up the volume up to 100 ml. While it is stirring, we have to prepare cotton flux for the flasks. For preparing cotton flux, you have to take one thick layer of sheet of cotton with your two hands, fold like this. Once the media dissolution is complete, we have to pour into the flask. We have to pour up to 100 ml. So we use only 1/3 of the place. Remaining space is empty. This is used for aeration purpose and also ensure proper autoclaving. In order to check whether the components are autoclaved or not, the media is autoclaved or not, we use sterility indicator. This is paper based sterility indicator. We have to paste onto the flask and we have to autoclave. If the autoclave is properly finished, then we will see the white strips turning into the black one. So this is the indication of the autoclave process is complete. Now the media components are completely dissolved. Now we have to pour into the flask. Cap the mouth with cotton plug and wrap with aluminum foil. Tie with the rubber band. Now this is ready for autoclaving. Once the media preparation is complete, we have to sterilize the media in order to use further applications. This is a typical autoclave where you can see temperature and pressure indicator, and these are the pressure knobs, and this one is quick pressure release knob. You can use when you are in a hurry, you have to use this one, but I will prefer not to use this one. Let it go on its way. We have to turn on the autoclave. So you can see here the bulb is glowing. Before keeping the media components to autoclave, make sure that the heater inside the autoclave submerged with water. Now I am going to keep the media components in the basket which we use for the autoclaving. Then keep this one inside the autoclave. While closing the autoclave, make sure that you are closing in opposite direction. Once the pressure and temperature reaches 121 degree Celsius and 15 lb pressure, you have to hold on that point for 20 minutes. Then you have to turn off the machine, let it cool down and remove the components. The same procedure you have to, while opening, you have to open in opposite direction. To conclude the video demonstration, we have discussed how to prepare bacterial culture media and how to prepare cotton plugs and autoclave it. During culture weighing up the media, we have to make sure that the components, media component should not be exposed to air, because those substances absorb the moisture and become liquid. So another thing is that for cotton plug preparation, we have to take a single layer of cotton, then we have to fold it. After autoclaving, we should not release pressure in a single shot. Let it go and come to normal pressure, then we have to open autoclave.
Now, I'm sure you have understood the how you can be able to prepare the microbiology media. The procedure of preparing a microbiology media will be applicable to all the medias where you are actually going to do the autoclaving, right? So the other class is, so when we talk about the eukaryotic media, the simplest eukaryote is the yeast, right? So these are the some of the media what you can actually be able to use for the yeast propagations. So where you have the CSM media, you have a YPD, a broth, we have the YP-gal, and you also have the standard minimal media where you are actually going to have the. And as far as the composition is concerned, you for the CSM media, you are actually going to have the CSM 0.74 grams. Then you have the yeast nitrogen base, which is 6.6 grams, and you are going to have the ammonium sulfate, which is going to be 5 grams. You have to have a glucose as a carbon source for 20 grams, and then you are also going to have the agar, which is going to be a gelling agent. And this is you are going to use for making the agar plate, which is going to be used for the growth of the Saccharomyces cerevisiae and for preparing the competent cells. Similarly, we have the YPD broth where you're going to have the 10 grams yeast extract, 20 grams of bactopeptone and 20 grams of dextrose. And this is commonly being used as a used media for the maintenance and the propagation of the Pichia pectoris and the S. cerevisiae. Similarly, we have the YP-gal. YP-gal is actually going to be used for is a standing media for the S. cerevisiae omitting the glucose repressions, and it is going to have the 10 grams bacto yeast extract, 20 grams of bactopeptones, 100 ml of 20% galactose, and then it's also going to have 50 grams of agar. Then we have the standard minimal media or SD media, and there you are going to have the 6.57 grams of yeast nitrogen bases with ammonium sulfate and without amino acids and sugar. And this is required for, so you're actually going to add these, amino acids and nucleotides at a growth of 50 microgram per ml. And this is the base media for the preparation of minimal and synthetically defined yeast media. And how you're going to prepare, you are just going to the same procedure what we have discussed for the microbiology media. You are actually going to take the media components, dissolve it into the 950 ml of water, and then you autoclave. Autoclave allow the media to cool down to 56 degrees Celsius, and then you add the 50 ml of filter sterile 40% glucose so that the final concentration becomes 2%, and adjust the volume to 1 liter if necessary. Remember that the glucose is very susceptible for degradation or for charring actually. So we don't add the glucose and then we do the autoclaving. We have to use the filter sterile glucose separately, and we have to prepare all other components into from the yeast media, and then we have to add according to our requirements. This is true for the not only for glucose but all other small nutrients like nucleotides, amino acids, and all that, right? So all these, the temperature sensitive chemicals we should, or the constituents we should add separately by doing the filter sterilization. Now let's move on to the slightly complex media. So that is the mammalian cell culture media. So I have taken an example of the DMEM media, and there are so many media available for mammalian cell culture. You have the RPMI media, you have DMEM media, you have MEM media, and so on, right? So when you want to make the DMEM media, you are actually going to take the DMEM as 13.4 grams per liter. Then you are going to add the sodium bicarbonate, which is from the 3.7 grams per liter. Then you are going to add the FBS, which is 10%, and you are going to add the antibiotic, which is the 1%. You are not going to use or put the BS and the antibiotic in the beginning. So how you're going to prepare the cell culture media? So add 13.4 grams of dry powder into the water and mix it to dissolve completely. Then you add the 3.7 grams of sodium bicarbonate, mix completely and adjust the pH to 6.9 to 7.1 using the NaOH or NaCl. Finally, add cell culture grade water to media to bring it to the final volume. Sterilize the solution using a sterilized membrane filter with a pore size of 0.22 micron. Remember, all the cell culture media you are going to use the filter sterilization with the help of the 0.22 micron. Then you add the supplements such as antibiotics and a serum, and then you can be able to sterilize using the aseptic conditions. Since this is a very complicated process, I thought why not we should take you to my lab where the students are actually going to explain you how you are going to prepare the media, how you are going to assemble the filtration unit, and how you are going to prepare the filter style media.
In this video, we will be demonstrating how to prepare mammalian cell culture media. For that purpose, we need basal media, which is DMEM, tuberculous modified Eagles media F12 high glucose. And we need FBS, fetal bovine serum. And we need antibiotic cocktail comprised of streptomycin and ampicillin. The basal media provides inorganic materials, amino acids, which are required for basic development of cell. And the FBS is used for providing growth factors to the cell. We cannot autoclave this media because it might degrade the components of the media. For that purpose, we use 0.22 micron filters. This is a 250 ml bottle top filter. Now I will be demonstrating how to prepare filters for media. We have to pack it closely so that it doesn't allow any leakage, and after this we have to put it for autoclaving. This is an autoclavable bottle top filter. After we have packed the filter, we have to keep it for autoclaving. For that purpose, we use indicator to check whether our filter has been autoclaved or not. When the lines on this strip turns black, it means that the filter has been autoclaved. In order to prepare media, now we will be adding the basal media to the already autoclaved double distilled water. We can use double distilled water or molecular water for that purpose. After adding media, we need to stir it on a magnetic stirrer for the components to dissolve completely. We can either use double distilled water or molecular water, but double distilled water is more preferable as it contains more ions than molecular water. After the media components have dissolved completely, we need to set the pH of the media. For that purpose, either we can use pH meter or pH strips. In this case, we cannot use pH meter as the bulb of the pH is sensitive to the media components and may get corroded. After the media components has been dissolved completely, we will be able to set the pH of the media. After the components of the media have been dissolved completely, we now need to adjust the pH of the media. The bright red color indicates that the pH of the media is in the range of 7.2 to 7.4. If the color of the media turns purple, then it indicates that the media is basic. If the color of the media turns yellow, then it indicates that the media has become acidic. Now we will be checking whether our media falls in the range of 7.2 to 7.4. After the media has been set, we now need to filter the media inside the biosafety cabinet as we have added the constituents in their non-aseptic condition. After the media components has been completely dissolved and the pH has been set, we now need to sterilize the media using membrane filter media. For that purpose, we use class 2 biosafety cabinets which are used for handling mammalian cell cultures. So this is a typical biosafety cabinet in which we perform the filtration for media. This is the control panel which is used to operate this machine. This is the on and off switch. This is the switch for normal light. This is the switch for UV light. Now I will be demonstrating how to filter the media. Now we are going to filter the media. For that purpose, we need a suction pump which can be connected to the bottle top filter. This suction pump is for the purpose of extracting the air from the bottle top filter so that we can filter the media. Initially, we need to check the media. We need to check the bottle top filter with less media to check whether if there is any leak or not. For that purpose, we are going to add around 50 to 100 ml of media. As we can see that there is no leakage in the filter, we can proceed with the filtration. After the media has been filtered, we now need to add FPS and antibiotic in order to make it complete media. The complete media comprises of serum, whereas the incomplete media does not contain serum. We are adding 100 ml of fetal bovine serum in order to make it 10% FPS containing serum. With this, we have prepared 1 ml of DMEM complete media comprising of 10% FPS and 1% antibiotic solution. So far, we have seen how to prepare cell culture media for mammalian cells. Although there are some precautions to be followed, like when we are setting the pH of the media, we need to use pH strips instead of using the pH meter. There are some components in the media that can clog onto the bulb of the pH meter and reduce its efficiency. Secondly, when we use the media, we need to thaw the media from 4 degree to 27 degree. But we need to thaw it first to room temperature and then to 27 degree to avoid change in the pH of the media. And also, if we are producing the media in larger quantities, we have to aliquot as per our requirements and then use in order to avoid contamination and change in pH of the media. Thanks for watching the video.
Once you prepare the filter sterile media, you are actually going to check, or you are actually going to take the different types of precautions, so that you should not actually prepare a media which is good for the propagation of the cell culture or propagation of the mammalian cells. So what you are going to do is you are going to take care of the pH. So pH should be between 6.9 to 7.4, and slightly acidic pH or slightly basic pH is going to destroy the cells. So if it is going to beyond this, it is going to do. Then you also have to do the filtration at a very low speed. So you should not do a very fast filtration, because if you do the fast filtration, it is actually going to compromise the pore size of the membrane what you are using for the filtration, and that may actually be able to responsible for bringing the bacteria what is present in the air, right? And that's how it is actually going to contaminate. Then your serum what you are going to use, the serum has to be heat inactivated, so that it should actually destroy the complements and other kinds of factors, so that it should not kill your cells, right? Remember that the serum is actually having a lot of immunologically active molecules, so you should do the serum heat inactivation, so that you should be able to remove these immunologically active molecules. Then you have to add the antibiotics after you are done with all these filtrations, and then you are going to add the sterile antibiotics. Once you are prepared the media, you are going to use the contamination checking, right? Because it has to be done before you add this media to the mammalian cells. How you are going to do that? You are actually going to take a 10 mm dish, right? And you are just going to add the 10 ml of your media, right? You're going to just take the 10 ml media and put it into here, right? And then you have to cover this with a lid, right? And then put it into the incubator for 24 hours. So then you put it into the incubator and let it be remain there for 24 hours at 37 degrees Celsius. And if you do so, if there will be a problem in your filtration, if there will be a problem in your serum and all those kind of things, you will actually going to see that the bacteria is actually propagating in your media. If the bacteria is not propagating for 24 hours or 48 hours, then that means that the media what you have prepared is sterile and the media is good for your mammalian cell culture propagation. So this is the DMEM media what you are going to prepare. Remember that the media has to be prepared in a small aliquot, so that if there will be a problem, if there will be a contamination, it should not spoil your media, actually the complete media, right? So you should not have like prepare 1 liter media. Instead you can actually have the 4 aliquots of 250 ml media, so that even if one aliquot is going to be contaminated, you can discard that and use the fresh one. Now let's move on to the insect cell line. So insect cell lines are also requiring the special media. So you actually require the different types of like Grace insect media, you require the Hinks TMT and insect media, insect IPL 41 insect media, then you require the TC100 and all that, right? And the insect media is being used for propagating the insect cell line, and these insect cell lines are being used for propagating the baculovirus, and that's how you are actually going to use that for protein production. So these are the compositions of these media and these are the applications. So for example, the Grace insect media is being used for the growth of the Spodortra cell line or SF9 and SF21 cell lines. Then you also going to have the Hinks insect media, which is going to be used for the culture of the cabbage looper or the Trichopilacia nil cells. Then you also require this media, which is going to be used for the SF9 and SF21 cells. And then you are going to use the TC100 media, which is for the production of baculovirus. And then you're also going to use these media for the mosquito cell lines, and especially the Aedes. And then you also require the Shadis Drosophila media, which is going to be used for the in vitro culture of the Drosophila minor clusters. So these are the some of the cell culture media what you are going to use for propagation of the microbiology media, like you are going to use for the growth of the the prokaryotic cells, and then you can actually be able to use the some of the eukaryotic media for propagating the yeast culture media and mammalian cell culture media or the insect cell lines. So with this, I would like to conclude my lecture. Thank you.